So in this chapter, 7.1 and 7.2 in your class, we're going to talk about the membrane structure. And the first part, we're going to talk about the lipid bilayer, composition structure, membrane proteins, and the movement of the proteins. So functions, you all know, remember that. These are very straightforward. These are the pictures that you're seeing for ages. And we'll stop right here. The first thing is that the lipids are amphiphilic. Now remember that this is one of the things that you definitely need to note down. And sometimes I say that many of the other things sometimes I extra say, but this is definitely important. Instead of amphiphilic, you can have the word amphipathic. Both means the same thing. Philic means love. You just both side actually likes. On the other hand, both side actually fear, phobic. That's basically the same thing. So it could be ampiphilic, ampiphobic. So essentially, you have a piece. Each end actually does something different. So that's what it is. One side is doing something. Say, for example, this is kind of a plus. If this is a kind of a minus, this is ampiphilic, both sides. If you do not have that, and if you think that the plus is philic, then it's ampiphilic only. If you think it is minus, then it is phobic only. Philic only, phobic only. But if you have both of them plus minus something like this, then it is ampiphilic. You can be ampiphobic as well. So the head group is polar, which could be either charged or uncharged, but it's polar. Polar means that it will always interact with a polar solvent. Water is the classic example. Polar solvent, water. So because why it is polar, that if I'm not going into the detail of that, but this is your very basic chemistry that you have H, O, and H. You have a little bit of charge distribution because oxygen is very highly negative. Hydrogen is just only positive, but all the electrons when shared, oxygen gives you most of the electrons. So it's slightly more negative towards the oxygen, slightly more positive. So this gives you the polarity of water. So water is a dipole, and it will interact with the polar head, but it will repel the hydrophobic tail. So it will not interact with that. So that's the structure of that. And the three classes of lipid will be your phospholipid, sphingolipid, and the sterols. So if we just look at the phospholipid, and you can easily see right here that you have a choline, then you have a phosphate. That's the reason it is phospholipid. Glycerol attached to two fatty acid chains. And these fatty acid chains are long carbon, you can easily see that right here, long carbon chains, and they are named as C14, 15, 16, depending upon the number of carbon you have that on that fatty acid chain. So that's how you actually name those. Many, many different names. And one of the important thing that what you are seeing right here, that if you actually do have no double bond, you actually don't have any double bond right here. All of them are single in between the carbon. We call them as the saturated fatty acid. Saturated fatty acid, no double bonds. If you have a double bond, then we call them as the unsaturated fatty acid, which is the next picture. You can easily see that saturated, no double bond, unsaturated, you have a double bond. So what happens right here, this double bonds actually allows that chain to flip, just like this right here, or kink. Not flip is not the right word, kink. It just can go in a different direction, whereas the saturated ones are very straight. So what happens, what the difference is, the packing of these lipid molecules next to each other depends upon whether their tails are straight or not. Straight will pack densely, whereas if you actually have this kink, they will not pack densely. And that is a hugely important biophysical property of the lipids which contribute to the cell. So very, very important. And we'll come to that. So we'll discuss that part again. So mainly three phospholipids. You have a phosphatidylethanolamine and then a phosphatidylcholine. And if you look at these structure, and again, remember that I will not ask you to write these structure or anything like that at all. This is not a chemistry class. This is just an illustration that this is actually a neutral phospholipid. This is a neutral phospholipid, but it's polar. but remember that. Polar but neutral. Why? Because you have a plus and minus actually for PE and PC. They cancel each other. So there is no net charge. On the other hand, see that for the phosphatidylserine, you have an extra minus. So it is negatively charged. So this is a charged phospholipid with a negative charge. These are neutral phospholipids without any charge, but all of them are polar, means they will, polar means like the head group is polar. Remember that the tail is always hydrophobic and non-polar. So this will actually face either the environment outside the cell or the cytosol that you actually are going to see. So here, another phospholipid, this doesn't have any structural role at all. It is present on the plasma membrane, but it does not have any structural role. This is your phosphatidylinositol, which actually always, as it is a phospholipid, you will have this resident phosphate all the time. This phosphates actually bind this 6-member ring right here, the inositol, to the fatty acid chain. But what happened that their phosphorylation status can vary. So the regular PI, phosphatidylinositol, can be phosphorylated at the fourth carbon. You do not need to know that, but just one phosphate at the fourth carbon. You calculate that like this: 1, 2, 3, 4, 5, 6. So that's how you this is organic chemistry, not needed at all. But you can add a phosphate, and which makes it a PI4 phosphate, or we call it PIP. That's how it is known. Then that PIP can be again phosphorylated with a PIP kinase to a PI4-5. So you can see that, right, this was 4, now you have added another phosphate to 5. You can actually add another one as well, not shown here. So you can have all these different phosphorylation status, PIP1, or just PIP, PIP2, even PIP3. So these molecules are essential for signal transduction, and we'll study that, and very, very essential. Doesn't have any architectural role. All right, the next type of lipid is the sphingolipid, and this could be of 2 different types. So the tail part right here, you can see that this is the tail, which is called the sphingosine. The head, which is the polar part right here, it could be either of the 2. Here, it is attached with the sphingomyelin. You can see that it's attached with the sphingomyelin, but you can replace that with this glucocerebroside. So this glucocerebroside can be attached right here, and it will be a different sphingolipid. So how this is actually named, you can probably correlate that the neuron's axons, I talked about that in the video, that the neuron's axon, the delivery part, the long part which comes out of a nerve cell neuron, is covered by an insulating sheath which is called a myelin sheath. And this myelin is the word that comes from here. So that myelin sheath, which is the insulating part of the axon, this is one of the lipid that is actually highly present in there. So that's it. Sphingolipid, 2 types, sphingomyelin and glucocerebrosides. The last part is the sterol, and the sterols, don't always think that the cholesterol is the only sterol part. And again, I talked about that, but not going to ask. The lower eukaryotes, like, for example, yeast. Yeast actually do have something which is not cholesterol called ergosterol. So this is a unique sterol that is present in their plasma membrane, ergosterol, E-R-G-O, then sterol. Then plants have something called stigmasterol. Stigmasterol, just like that. Here it is cholesterol, and it's a immensely important lipid, which actually does have, again, a polar group, which is this hydroxyl. Right here is polar. And then you have this planar ring-like structure and that tail, and that total part is completely hydrophobic as well. Not only it is important for the membrane, most of your hormones, which does have the word estrogen, progesterone, testosterone, all of them are actually derived from the modification of cholesterol as well. But it is immensely important in the membrane structure, and we are going to see that how this actually influences the membrane rigidity. Very, very important. So here, something we are going to talk about. So why nature actually chose lipid to be the plasma membrane. I mean, like, you have carbohydrate, you have proteins, they could have made the plasma membrane as well. The reason that we can actually see that why they chose, if you actually take right here a lipid which does have a polar group and just have a single fatty acid tail right here, you actually make a van der Waals envelope or a shape like that, like a little bit conical at the bottom. If you actually do have two of them, you actually make a kind of a shape with a van der Waals force. Again, remember that your van der Waals envelope or force is basically a weak interaction, very, very weak atomic force interaction, even weaker than hydrogen bonds. But it is an interaction which can attract or repel molecules. So that van der Waals force, due to these type of structures, the lipids, when you disperse any kind of lipid, it's going to automatically form something. Classic example that you will believe that you just take a glass of, like half a glass of water, just put a little bit of oil on top of that or in a bottle, just shake it and leave it alone. What happens? You will have a clear layer of lipid at the top. You shake it, they all disperse, but they will all come back and form a layer. That lipid layer is no different than this structure that you are seeing, which is, that's why it falls asleep. So there is no difference in between that, which is this structure. So lipid layer is that. So the beauty of that in the aqueous environment, these molecules being amphiphilic or amphipathic, they self-assemble. So that's the most important word, that the lipid molecules which are amphipathic or amphiphilic self-assemble, and that's the biggest property that they can actually make an envelope. You cannot have that property from carbohydrates or the proteins. So that's the reason that automatically nature chose lipid to be the membrane. So what is the difference between the micelle and the bilayer sheet? You can see that this is another structure where you actually have a globular structure where the hydrophobic core is towards the inside, and this is a droplet. So think about this, that when you actually take some oil and water mixture, when you shake it, then all these tiny droplets that you can see, those will be in the micellar form. And when they coalesce together to form a layer, they will be in the bilayer form. So this is basically the difference in between that. So if you take probably a microscope at that level that you can do, you cannot technically do that even under electron microscope, but you can actually see that how these micelles are actually formed. So this is the same thing. So here is a classic experiment, and that how that the membrane is actually studied. So you actually can take just a membrane, and you technically you can do this experiment with the material that you have in your garage. Literally, you can actually just get a little bit of meat and then chop it up a little bit, and you actually just put some acetone in that, like in a ground meat or something like that. What you are going to do, that you are going to dissolve all these lipids in that acetone, and then just use a fine filter or cheesecloth to filter that material out. Now you just let it on a flat dish or something like that. What's going to happen, that the protein is actually going to get precipitated at the bottom when you are filtering, so it's not going to pass. What is dissolved in the acetone is the phospholipid only from that meat product, and you let the acetone evaporate. So once the acetone evaporates, you can take those phospholipid, and if you actually add them in a water back again, you will find a structure which we call as a liposome. So they will naturally form together a micellar structure, but many times when they are all separate apart, they will also may have a hydrophilic core. So little bit difference between the micelle. Let's see that the micellar structure, you have completely hydrophobic core. On the other hand, here, the liposomes, you actually do have an inner aqueous compartment as well. So you can spontaneously form this, and think about that. This is your tiny kind of a cellular structure that you have. Think about that this is your plasma membrane, and that is your artificial cell. And the artificial cell that has been created is exactly this way. You put your self-replicating genomic machinery in there, and this will actually start behaving as a cell. So you can do that. On the other hand, in this experiment, if you actually partition these two with some tiny holes in there, in this partition, and you just add the phospholipids on each side, it will automatically, around that small hole, form a lipid bilayer. So this is the self-assembling property, and that's the reason that we actually use phospholipid for the cells. All right. So here another thing, that biological monolayers. Why we have bilayers and why we have monolayers. We talked about that not always, but there are some cases that these biological monolayers are present in archaea. These are called extremophiles. That means they are in very hot environment, usually chemically very, very demanding condition, hot water springs, hydrothermal vents. You actually find these archaea. They are not bacteria. Remember that bacteria is an entirely different domain. These are prokaryotes, but bacteria is different, archaea is different, and then you have a eukarya. So different domain because they differ significantly than the bacteria. They have these monolayers, and just a modification. Think about that. You have a bilayer, and if you have the polar head group on this side, you have a polar head group on this side. And if these individual molecules are covalently joined in the middle, you actually get this. And this is how it is actually, these membranes arise actually that way. And why it is actually done, it behaves as a phospholipid bilayer, but the reason that in the extreme environment, to maintain the integrity of the membrane, these bilayers will fall apart in that crucial that temperature. So that's the reason that they could keep the membrane integrity. They actually evolved to form these monolayers. So that's one of the things. We actually find monolayers in our cell body all the time as well. So the LDL or any of those that we actually see, low density lipoproteins or high density lipoproteins, these are cholesterol movers. Remember that you cannot move, say, cholesterol through the blood from one place to other. And the problem is that if water and oil doesn't mix. So if you just put, like, really in the middle, you have a cholesterol right here, it's going to immediately stick onto the side of your blood vessels and form a plaque. And that actually in reality happens, and we'll come to that. So how you transport the cholesterol in and out of the cell? So they are wrapped around by a single layer of this phospholipid monolayer. And if you think about that, your phospholipid monolayer do have an hydrophilic or polar head that will interact perfectly with water. Usually it's attached with this ApoB protein, and that makes it a lipoprotein. And in this condition, the LDL or the HDL can be transported happily into the bloodstream. But the question that why high cholesterol or these actually is a problem and what happens. While being transported, this is entirely different. This is you don't need. Sometimes these layers can get disrupted or the proteins may fall off. At that time, this can happen in real time in the arteries or the veins or in the capillaries. At that time, that cholesterol right here in the middle, they are just directly contact with the water, and they will actually precipitate on the side of the blood vessels, and clogging or plaque formation happens this way. So it's just that mechanically, while being transported, this actually happened. So here is how this is actually formed. The cholesterol or the triglycerides are formed from the endoplasmic reticular membrane. And this is a typical called a lens formation, that how these monolayers are formed. And these are the transportation of your triglyceride and cholesterol in your body. Classic example of these. All right, first clicker question. I think I got this one right today, hopefully. And I don't have that answer. Which of the following is involved in signal transduction? P, PC, PS, and PI. 15 seconds. One minute, stopping it now. All right. All right. So phosphatidylinositol, that is the correct answer that you can see. Yeah, a couple of you answered ethanolamine. Remember that the inositol, the PI, is the only one which could be phosphorylated in different states. That's the only molecule that can be used as the phosphatidylate. So 4, phosphatidylinositol, is the correct answer. All right. So next we are talking about that all these lipids that we talked about, they can move in the like literally they dance around in the plasma membrane, like they are just having a party there. So I'm like, literally it's kind of it's funny, but that's what it is. They can have lateral movement, they can rotate, they can flex their tails, and even flip. So why that's not a party? So you need a flipase enzyme to do that. So here, one of the most important, all the jokes apart, what is important right here to remember that the lateral diffusion, this is your take-home message from this, the lateral diffusion, flexion, or the rotation, none of these actually require any kind of cellular energy. This is completely dependent upon the environmental condition, that whether you are in hot, cold, whatever physiological condition, they do not need any cellular energy. Whereas the flip-flop, which need a flipase enzyme, also important thing, that lateral diffusion, flexion, rotation doesn't need any enzymatic intervention at all. It is just a natural way of how the lipids work. Whereas the lipids, where it need the flipase, this is an extremely energy, it's a demanding process which need energy. So you will need a lot of ATP to do that, comes from the cell. Why it is so, remember that you have this, the polar head group. This polar head group have to actually travel through this hydrophobic core onto the other side, which is not a favorable interaction. It is energetically not favored. You just cannot. So basically think about that if you have an oil layer and water underneath, the water by itself cannot penetrate through that oil layer and go by itself until it is forced by something else. So you can just poke it and then it can allow, but you are just putting external energy. So that is actually required to do that. So that's the important, is energetically not favorable, need an energy, need an enzyme. That's your take home. So here, the membrane fluidity actually depends upon the temperature, and it's temperature dependent, and see that's how it happens. And a critical temperature, that what you have, what you call as a transition temperature of the biological membrane. That's a very important concept. And what is a transition temperature? A transition temperature is a particular temperature below which the plasma membrane will actually have a gel-like consistency, thicker. And you can see that the fatty acid tails are straight at this time, and the membrane is actually thicker. So it actually varies in the thickness. The membrane is about 3 to 6 nanometer. Why? It is a range, not 5 nanometers exact. Because the membrane can actually become thinner or thicker depending upon that. And this is actually the classic example that it is thicker. On the other hand, above the transition temperature, all these fatty acid chain actually get disrupted, and this is just like random motion. This is a random motion, and they start vibrating motion, and that packs these two layers much closer, and this actually gives you a fluid-like consistency, and at this temperature, the width of the plasma membrane is actually much less. So this is a very classic example. So here, this actually gives you another view of that we just talked, just reinforces the idea that what happens that the short fatty acid tails are actually easier to melt and they are more fluid. So if you actually need an area where you actually need a more fluid membrane, so you need a more fluid membrane where you have a particular tissue where you need a huge amount of material exchange for that process. You need a more fluid-like environment so that all the proteins there can move. You need a more fluid environment. So those areas actually have a short fatty acid tails. So what do we mean by the short fatty acid tails? Maybe they are 12, 14 carbon, like this. On the other hand, where you have a long fatty acid tails, which is say 17 carbon, 18 carbon, up to 24 carbons. Those are long fatty acid tails, and they will actually pack the membrane, make them much more rigid. And another biggest advantage that we said, that as soon as you have an unsaturation or a double bond, is going to kink that, and that will not pack the lipids completely. Whereas if you have a saturated fatty acid, only single bonds, they will pack these lipid tails so dense that it will be less, you will have a less fluid condition in this condition. So here is a classic experiment which actually shows you the length of a fatty acid tail right here. You can see the length. So this is actually the number of carbons, 12 all the way to 24, then 18. And right here, you can see that if you have a zero double bonds, that means up to this 24, 12 to 24, all of these are saturated fatty acid. And think about this. Transition temperature goes from -1 all the way up to 80 degrees with this. See that if you have an unsaturated fatty acid, saturated fatty acid without any double bond, its transition temperature is 55 degrees. Now what you do, that the same place, you just introduced one double bond. See that transition temperature drops 54 degrees. So it is hugely dramatic that just one double bond can drop the transition temperature from 55 to 1 degrees to another 52 degrees. Then it is not so much, but just one or, so the emphasis is just one or two double bonds. You do not need a lot of double bonds, but just one or two double bonds absolutely reduce the transition temperature. So if we actually think about this, what the take-home message from here, you can see that as you increase the chain length, the transition temperature varies to some extent. But introduction of a double bond, it varies dramatically. So if we just think about that, which property, saturation or length, contributes to the more, it is always the saturation. That's your question. That saturation is always more responsible for the change in the transition temperature. Length do some contribute, but that comes always second after the saturation. So very important concept. You can always think about some critical question from this one. So here also another very important concept as well. I think that these two are the most important conceptually idea. Rest, most of it is kind of self-explanatory. That the membrane fluidity is actually influenced by the sterol content, which is here is a cholesterol, where the red part is the polar group and the rest, the planar and the tail, these are all the hydrophobic part. So what happens that you can easily see, the question is that what cholesterol actually brings to these membranes. So if you just go to a kind of like a 101 biology book, always you will see that there's a word always written as that cholesterol increase membrane rigidity. So that's the word, that sentence that you see all the time. Cholesterol increase membrane rigidity. So this is correct and false both. So it is actually, when you go into the detail, it is not true actually. So this is the membrane shape. Don't worry about this. This is very self-explanatory. But here is the thing. That does cholesterol make the membrane more fluid or less fluid? So here is a direct quote from this book, that at physiological temperature 37, cholesterol decrease the membrane fluidity. That means, yeah, it makes the membrane rigid at 37 degree. But that is the only thing that is actually written in all of those 101 biological sciences book. What happens that at lower temperature, cholesterol make the membrane more fluid than the phospholipids, and therefore it is temperature dependent. You cannot, so at this level, when you actually study this, you are not going to bound by the idea cholesterol increase membrane rigidity, because it is not correct. Cholesterol, actually the rigidity of the membrane depends upon the temperature at which cholesterol is. So it can does both. It can increase membrane rigidity, it can decrease membrane rigidity, but it does that in a temperature dependent fashion. So this is another very important concept for your exam quiz everywhere. So this is the classic example. This is not in your thing. So this gives you an idea what we mean by this. So this is the oil that you change in your car. And it always comes as like a 5W40, 10W30, 5W30. And what does this mean? It just means the same way cholesterol that does that on the membrane. So what happens that so if you take a 0W30 oil in there, you can see that as you lower the temperature, it tries to make the oil more fluid so that it actually can serve the purpose that it is not getting thicker. So that's the idea. It's a 0W30. These are all multi-weight oils. On the other hand, see that 20-weight W30, it can just go all the way up to 0 degrees. After that, that oil will be too thick. And cholesterol exactly serve this purpose so that it can goes to a lower temperature and you can survive in the cold that so when you're exposed to minus 10 degrees or going out in the snow, so the membrane just underneath you have a 20, 30, 300 layers of dead cells on the top. But after that, you have some live cells and you do not actually get it frozen. But yeah, of course, you can get frostbite. That means when you get a frostbite, the temperature goes below that, even that oil cannot save you and the cholesterol cannot save you, and that's where it actually starts freezing. So this is just kind of an idea that how this multi-weight oil and cholesterol is the same kind of thing for your membrane. So on the other hand, all the biological membrane, think about that. If you make a liposome artificially, everywhere it is the same. But the biological membranes are absolutely asymmetric. What we mean by the asymmetry, that there are some certain type of proteins on one side and certain types of lipids on one side. There are certain types of lipids on the other side. So you can easily see that there is a distinct asymmetry. Inside the membrane, there are lots of these phosphatidyl, you can see a phosphatidylinositol, ethanolamine, a lot of them. But you can see that there is a lot of these phosphatidylserine where you have these negative charges right there, lot of them. On the other hand, you can see that on the other side, the outside, you don't have these negatively charged lipids that much. So the inside of the cell is always negatively charged, and it has something to do with the potential of the plasma membrane. And it has to, again, this part is not needed, but always your potential difference outside the plasma membrane and inside is about minus 70 to minus 90 millivolt. And that is maintained by the different proteins and the lipids, which gives this kind of asymmetry. And this is required for all these physiological functions. So that's the take-home message, that they are asymmetric. So we have seen that already, and how this asymmetry is actually maintained. So what happens that if you actually have a cellular organelle where you have the lumen, think about that. That lumen is actually basically the exoplasmic phase of the plasma membrane. So this is the environment, and the lumen is actually the environment. So if you see that this is actually a transmembrane protein, where this portion, these two tiny small spheres are facing the lumen. Suppose this is a secretory protein, and due to exocytosis, this is actually coalescing with the plasma membrane and secreted outside. And when it secretes outside or just goes out, this exoplasmic segment is going to face the outside. So this inner portion is the outside. So this just gives you an idea that the membrane asymmetry is always maintained and it is conserved. And here in all different places. Clicker question 2. Which property contributes more to the membrane's fluidity? Transition temperature of the fluid transition temperature of membranes, degree of protein saturation, number of fatty acid bond saturation, length of the fatty acid chains, all of the above. Make sure your pointer is which property contributes the more. That's the more, that word is the most important one. $15 seconds. All right, stopping poll now. Stopping now. Yep, that is the correct answer. So number of fatty acid bond saturation, we just saw that just one double bond dropped the temperature that much. So that is the correct answer. Protein saturation is presence of protein. I mean, like, it's a weird word there. Length of fatty acid chain is much less. So, all right, so that's good. Let's go to the next one. Cholesterol make the biological membrane rigid, true or false? $15 seconds. Stopping it now. Good, everyone. Stopping it now. Yeah, of course it is false. So I mean, like, so now you can spook all the freshmen taking biology. They ask this question to them and they say they are wrong. They will be completely confused. So you can easily do that. So I mean, like, you can tell only big kids know that. So all right, the second part, membrane proteins structure the basic function, and here we are going to look at these different proteins. So we talked about the lipids, now the proteins, and again we are going to give you just some examples, and we're just going to your question will be that you are showing us that many different types of proteins and what we are supposed to know. We'll come to that. So you have these integral proteins, which are also called the transmembrane proteins. Remember that, that's your basic. Integral, transmembrane, same thing. Then you have the peripheral protein, and there's other proteins which is called the lipid-anchored protein. So we are going to talk each one of them. So here, what you see that the first type of transmembrane or the integral protein, they actually pass through and the membrane, and the middle portion, which is through the domain, which is through the lipid bilayer, this naturally form alpha helices. So these are called the hydrophobic part. These are the alpha helices. And one of the property that the amino acids present in that domain has to have. So if you just remember from your biochemistry, the there are many different ways that you can classify the 20 amino acid. They could be polar, they could be non-polar, they could be acidic, they could be basic, but they could be also hydrophobic or hydrophilic. So in order to pass through the core of the membrane, they have to be hydrophilic or phobic. Phobic? Everybody good? They have to be hydrophobic. So the hydrophobic amino acids are present in there. Particularly, there could be some hydrophilic or polar, but they has to be buried inside. They cannot be interacting with that. So mostly hydrophobic amino acids are in there so that they can actually stabilize by many different types of interaction. There is always a van der Waals interaction in between the lipid and the adjacent groups as well. But along with the van der Waals interaction, the hydrophobic interactions are also important. So two take-home message that all these transmembrane proteins actually depend upon two types of interaction with the surrounding lipids, van der Waals and hydrophobic interactions. So here is an example of a transmembrane protein, which is a glycophorin A. And again, here the question could be that the next few slides, there are a couple of different proteins. What you are going to ask? I'm not going to ask the function or where they are present about these proteins at all. Just giving an idea, an example that what are the different kinds available. And just one thing here, that a group of protein first that we are going to talk about, they actually pass like an alpha helices through the transmembrane portion. So here is about a 23 nucleotide pass, and it's a dimeric protein. You have a monomer here on one side, another monomer in one side. And that's the molecular structure. You can see that it's heavily glycosylated. Those things are not needed. But here, this is the portion that is actually passing through the transmembrane domain. And here, this is just a single pass. So what we mean by a single pass transmembrane, that the protein from the N-terminal to the C-terminal passes just only one time through the membrane. So, and this passes with like an alpha helix. So here are just a couple of different examples. So the left-hand side, what you're seeing is a bacterial rhodopsin. So rhodopsin is actually with the retinol is present in your rods and cones in your retina. They actually capture the light, and this is a bacterial homologue of our eukaryotic rhodopsin, which is a little bit different. So this retinol actually absorbs light energy, and then it evolves into a proton pump. So that's the, and you can see that right here. You have, this is a 7-pass. That is not needed, but I'm just saying this is a 7-pass transmembrane, and all these alpha helices pass through that core. You can easily see that. On the other hand, right here, this is a glycerol channel from E. coli. These are from bacteria. This is a glycerol channel, and these are solved structure. So these are solved atomic structures, and these are the two glycerol channels in two. And here is kind of an interesting example that what you see, that here you can see half of these helices is inserted through the hydrophobic core, and here half of the helices is inserted to halfway through. On the other hand, you can see here it goes kind of a straight, and then you have a kink here as well. And overall, if you see that these alpha helices are kind of a straight, whereas these are in a kind of an oblique shape. So just trying to give you an idea that they come in all shapes and sizes. So that's the take-home message. You do not have to remember that there is a break to helices, which is straight, which is round. You do not need. Just have an idea. Another classic example, this is a photosynthetic reaction center from Rhodopseudomonas, a photosynthetic bacteria, and 11-pass transmembrane protein. So you can appreciate that how complicated these structures could be. This is also a proton pump. This is the second class of the transmembrane proteins. So this is what you need to know. The first set, you needed to know that those pass through the membrane with the alpha helices. This is the second one. They actually made what is called a beta barrel. So the difference is that if you just remember from your biochemistry, these are the beta sheets. These are not alpha helices. And these beta sheets, here you have these 14 strands. These beta sheets actually make a barrel-like structure through the membrane. And these are actually called the porins in the membrane. Just think about that. I talked about that in the lecture, that if you just take a paper towel roll and stick through the membrane, this will be something like that. And the central core, this is usually aqueous, water filled, and there are some controlling structures in there. So usually these structures actually can control the diameter of the pore. They can either constrict or relax depending upon the molecule. So they will allow some molecule to pass, but not all. So that's a second type of transmembrane protein, which is made up of these beta strands. And here is an example. This is a porine from an E. coli, 16 beta strands. Just one example, nothing else needed. So here, why I put this picture from your textbook itself, that this is actually a solved structure of a porine molecule, and on the surface, the porine molecule is actually in the gray, in the background. But in the more darker gray or the blackish color with the red, that what you are seeing, these are your phospholipids. So you can see that each one of them has 2 fatty acid chains. Why this is a mind-boggling structure? Because if you actually go and look the internet, that solved structure, atomic structure, you will always see it is either protein or DNA RNA. You actually do not see carbohydrate or lipid structure in there. Why you do not see that? Because they are extremely floppy, move around. They don't have a rigidity like a 3-dimensional protein or DNA RNA. So hardly you see any structure of lipids or carbohydrates. But here, these are the molecular structure of the phospholipids, and why it is so that they are so tightly packed outside. This is artificially done. Remember that this protein is crystallized artificially. These phospholipids are added onto them artificially. This whole crystallized structure is then solved by an X-ray diffraction by this. But all these lipid structures are the reason for that. They are due to all these hydrophobic and van der Waals interaction. They pack around that beta barrel so tightly, they are extremely ordered. That's the take home. They're extremely ordered, and this gives you an idea that when these porins are actually in the membrane and passing through the membrane, how rigidly they are held by the membrane. They are not going anywhere. So this actually gives the insight that how you actually study these interactions. And again, always, as I said, no matter whether this is alpha helix or beta barrel, remember it is always 2 types of interaction, either van der Waals or hydrophobic interaction. So that means that all the proteins which are sticking out towards the outside, here is a very interesting idea from here. All the proteins that are sticking outside and interacting with the lipid molecules, they have to be hydrophobic, but not the other way. Think about that. You have a central channel, and if it is an aquaporine, water has to pass. So all the proteins, all the amino acids which are facing the inner surface has to be hydrophilic. So all hydrophilic amino acids are facing the lumen or the central channel, whereas the hydrophobic residues are actually towards the outside. So this is also a very important concept, that how physiologically proteins are responsible. So here what we are seeing, on the other hand, the lipid anchored proteins, the next type. There are 3 different types. You can have a GPI anchor. This is a very complicated one, whereas in an acylation or a prenylation. These are the 3 different types of lipid anchored proteins. And as the name suggests, many times these are commonly known as a proteolipid as such. Proteolipids, sometimes people call membrane lipoproteins. Both ways are correct, lipoprotein or proteolipids. These are the ones that people are talking about. So here in the acylation, that what you are seeing, that you have the protein right here, and it could be many different types. And it forms an amide bond with this long fatty acid chain. So this is a myristyl, which is a 14 carbon fatty acid, and it forms the bond, and that myristyl 14 carbon is actually inserted into the upper leaflet of the bilayer, and you form an amide bond. Here there is another different way the bond is formed. In the prenylation, this is a quiz question. The prenylation, the last residue of the C terminus where the bond is actually being formed is a very specific residue. See that there is a sulfur in there. So there is a very specific amino acid that needs to be present in all phenylated bonds. So what is that amino acid? Anyone? Cysteine, that's correct. That's the only one, cysteine, out of all your 20 amino acids, that's the only one contains the sulfur, and that is needed for the thioether bond formation. So always the C-terminal will have a cysteine, and this is called the isoprenyl group. And here this is an example of a farnesyl. This is a C15 anchor. That means you have this carb right here. And this, the difference between these two, the myristyl anchor is actually straight, although it's shown right here, but it's a myristyl anchor is a saturated fatty acid. Whereas the farnesyl, the C15 anchor, this is actually an unsaturated fatty acid. So they can come in all different types of different anchors. So prenylation and acylation. On the other hand, this is a very complicated one, and again, you do not have to memorize the organic structure or anything like that, but just remember that how this is actually being formed. Here, if we look at that, right here you have a phosphatidylinositol right here. And this phosphatidylinositol was actually the member of that signaling molecule. It is attached to a core polysaccharide, 3 mannose, and glucosamine. Then this is a phosphoethanolamine, which is attached to a protein. So you have a protein. This is a phosphoethanolamine, which is a head group of the one of the type of phospholipid. Then you have a mannose, and then you have a head group of another phospholipid. So a very complex type of protein, and this is what we call as a glycophosphatidylinositol, and this is called a GPI anchor in short. This is a complex one. So the peripheral membrane proteins are very easy, that they can come off the proteins, come off the surface of the lipids very easily. They can be removed. And here is an example of phospholipase A2. There are many phospholipases, and it just depends upon where they are going to break the bonds. So what happens here, that these phospholipases, they are peripheral proteins. When they actually attach onto the surface of the proteins, they actually have it. So the phospholipase do have a positive charge, and it docks into a particular area where you have a negative charge. And then what happens, the phospholipid actually moves into this hydrophobic core, which opens up upon this interaction. So again, repeating that. So this protein is moving around, and it docks onto the phospholipid, where you have a plus minus electrostatic interaction. When it docks to that certain area, a hydrophobic channel opens up due to a conformational change of the protein. When this hydrophobic channel opens up, a phospholipid actually goes inside that and binds to the active site where there's a calcium. So the calcium just actually orients that phospholipid, and then you can see right here, you have the phosphate right here, and this is the active site. And this coordination right here, the phospholipase will actually cleave that phosphate from the fatty acid. So the ester bond will be actually cleaved. So this is basically the function of the phospholipases, that they will actually so recycling of the membrane or membrane turnover, that's the main function for this. So this is another interesting. Many of these cytotoxic snake venoms, these are actually super rich in phospholipases. So basically what happens, that goes into your body, basically they dissolve the cells in the blood or anything. Remember that these are the viper venoms, not the cobras. Those are neurotoxin. But most of the viper venoms, they are loaded with these acid phospholipases. That's how they dissolve the membrane, by this mechanism, just breaking the phosphate and the fatty acid tails. All right, blood group antigens, really not important here. We are not going to talk about that at all. That all it says is that these are all attached with either lipid or the protein. And I'm not going to ask any of those galactose or any of those enzymes. That is beyond the scope of the biomembrane structure. But if you're just interested that how the different ABO groups work, that's an idea. So here we are going to talk about some of these ionic detergents and how you can use them to study membrane proteins. So you have two different types, ionic, non-ionic, very straightforward. And this is actually a better picture because this gives you an idea. They always have the hydrophobic tail, and they have this polar, either charged or polar uncharged non-ionic part. So you can easily see a detergent molecule is almost similar to a lipid molecule. And this is the reason that the detergent actually can solubilize these proteins, because what's going to happen that the lipid will dissolve or interact with the hydrophobic part. This part, which is the polar part, charged part, will interact with the water, washes away, and the way it actually forms, you can easily see right here that all these hydrophobic tail and the polar head group, they can actually dissolve either the lipid or they can dissolve the protein as well, because remember that the proteins the transmembrane domain of the proteins are hydrophobic. So this is a way that you can dissolve the membrane proteins to study using the detergents. So here is an important concept right here, that what is your critical micellar concentration. So if you actually do have a high concentration of the detergent, then you have a mixture of lipids as well as the proteins. You have both, mixture of those. Because you have lots of these molecules that will take out lots of lipids and they will form these micelles. So you have mixed. You will have some just only protein, just some of them could be only lipid, not shown in here, and some could be protein and lipid all. But there is a critical concentration, which is called the critical micelle concentration. If you actually make the detergent in such a way that there's not a lot of detergent molecule, they preferentially binds to this part and dissolves the protein first. And this is actually a very unique way that without any of these other molecules, so the mixed type, you can extract the protein by using the CMC of that particular Triton X100 or anything that what you are using. So this is a great way to purify these membrane proteins, and the membrane biologists do this all the time. Clicker question. There you go. Which of the following interactions contribute to the stabilization of the transmembrane domain of the integral membrane protein? Hydrophobic interaction, van der Waals force, hydrophilic interactions, A and B, all of the above. $15 seconds. Stopping it now. Stopping it now, right here. All right, very good. That's the correct answer. A and B is the correct answer. Good. All right, so the last part, we will go through this. So we have talked about this FRAP all the time, so we're just going to stop right here. The top part we have already seen before. Just the take-home message from here, see that this was the fluorescence before the bleaching. When you bleach, it completely drops down. It becomes white. And then you allow the fluorescence to return right here. But the important part is that you don't get the fluorescence back completely to the 100% level. And why it is so? Because all the proteins are not mobile. That's the take-home message from here. Very important that 50% mobile and 50% immobile. And that gives you the idea that all the proteins in the membrane is probably not moving. So how this is actually achieved by the cell. And again, remember that we talked about this epithelial cell all the time. These red proteins, protein A, is only on the apical surface. These are on the basolateral surface. And they cannot mix together Why? Because there's a tight junction around these in between the two cells, and that actually restricts the movement of these proteins. And this is a very important concept, that how these membranes actually help them to keep them in position. So tight junctions. And this is the last thing, that this is called the lipid raft. And then we'll have two clicker questions. The lipid raft is very rich in cholesterol. So this is rich in cholesterol, and you have specific lipids in here, which are the sphingolipids as well. And that part of the protein, you know that physiological pH, that part of the membrane is extremely rigid. And some specific proteins have high affinity for that. So if they have a high affinity, once they enter that lipid raft, they are not moving anywhere. So they are lodged into that area, and that's how they can perform a specific physiological function. So that's basically the overview of that, this idea that what are the lipid rafts, that cholesterol-rich sphingolipid-rich area, rigid in function, localized protein, don't allow them to move. And these are some of the ideas that how the different ways the cell can immobilize the proteins. This is very straightforward. Just gives you an idea that how, on the other hand, proteins can move in the membrane and self-assemble to a multimeric structure. All right, quiz questions. This is a little bit difficult, I guess. Nope, not that. Oh. The glucose transporter GLUT2, we saw that in the fluorescence picture, localized in the basolateral membrane of the epithelial cells of the small intestine. How do these cells achieve this polarized distribution of GLUT2 with the apical brush border cilia, tight junctions, desmosomes, hemidesmosomes, all of the above? $15 seconds. Stopping it now. Yep, that is the correct answer. Very good. And that's it, pretty much.